and he's a fantastic scientist now working in Dr. Oliver Hober's lab at Columbia University. And his research now is mostly on the sexual dimorphism in the circuitry of elegans. But today he's going to tell us about his PhD work on the aversive behavior. He comes from Dr. Zhimiao Pan's lab, which is one of the top elegans lab in Taiwan. And if any of the students in the audience who is interested in applying postdocs in the US, and John is definitely a good person to talk to and then get his experience. Without further ado, John, please take away.
Can everybody see my slide?
Perfect.
Okay, give me a second. Okay, never mind. Okay, so thank you for the nice introduction. First, I would like to thank you all for attending, and especially Vivian and the organizers for the invitations and the chance to share the recent published story that I have done in my PhD in the Penn Lab in Taiwan. So today, I'm going to tell you a neurophysiological basis of stress-induced aversive memory in nematode C. elegans. And before moving forward, I would like to especially thank Jen, who was a master's student in the Penn Lab and now is doing her PhD in Johns Hopkins, for repeating all my experiments to achieve the qualified replicase number for publication. So then let's begin. So it's evident across most circumstances, our behavior and responses to the stimuli are directed by our internal states. For instance, if you have been starved by doing complicated molecular biology or doing image on the confocal for the whole day, then you must have high intention to eat those that tasty food from Shake Shack. By contrast, if you've already had a meal or feel sick, the same food from Shake Shack definitely will not be as tempting as the previous scenario to you. So how do the internal states affect the behavior? One way it works is that internal states such as hunger, fullness, sickness are translated into cellular processes such as metabolic pathway, inflammation. And our nervous system then can sense the alteration of the cellular process, probably such as the insulin level or the inflammation status, for example, to adjust to the according behavior. And this behavioral modulation by internal state is evolutionarily conserved. So using model organisms, scientists aim to delineate the underlying neuronal mechanisms. So for example here, in the innate fruit fly, they are attracted by the odor of the pathogen bacteria. However, after ingesting the food with harmful bacteria, their preference change. They intend to avoid the food source with pathogen bacteria. How does this behavior change happen? It is shown that after injection of harmful bacteria, there was like an inflammation happening in their body, and an immune response is induced from the fat body and signals target to the brain to initiate an aversive memory. So here, to further understand how the aversive memory form at a cellular level, we use C. elegans as a model. So here, I would like to convince you that C. elegans is a good model for several reasons. First of all, it's genetically tractable, which means we could play a lot of genetic tricks to dissect the pretty detailed pathway or genetic interactions. Second, the connectome of the nervous system in both sexes are fully resolved in C. elegans. So this allows us to, look around the connectivities and to play around with the connectivities. And third, with available reagents we have, we can reveal the alterations nearly in single cell resolution. So a previous study from the Rufkin lab in Harvard, which I think it's in the same buildings as Jerry, showed that homeostatic imbalance in a worm changes worm's preference toward the innocuous bacterial food source. They hypothesized that during disruption of cellular homeostasis, such as DNA damage or mitochondrial stress, non-neuron tissue will release yet unidentified molecules that remodel the nervous systems for the behavior changes. Here, my previous lab are particularly interested in how mitochondrial stress reshape animals' behavior, because mitochondria is one of the crucial organelles to produce energies and to balance energy production. Moreover, several neurological or neurodegenerative diseases are linked to mitochondrial dysfunction. So the naive C. elegans is attracted to innocuous bacterial food source. And we hypothesize that with concurrent internal mitochondrial damage, the animals will establish an aversive memory toward the bacteria, probably via remodeling the nervous system and re-evaluated the bacterial cues. Therefore, as a result, the animals avoid the bacteria. Moreover, when a worm encounters the same bacterial cues later, even without mitochondrial stress, they will show avoidance behavior as a consequence of this aversive memory. So here, we first test whether animals' behavior toward innocuous bacteria are changed by concurrent mitochondrial stress. We induce mitochondrial stress in animals by treating them antimycin A, which is a drug that causes failure in mitochondrial electrotransport chain. We and others have also shown that this drug only induces mitochondrial disruption in the intestine and the muscle of the C. elegans, but not the nervous system. We found that animals develop this aversion towards the innocuous bacteria with concurrent mitochondrial stress, and the aversion becomes robust over time and shows significance as early as an hour after the treatment. We calculate this aversion by avoidance index, which is the ratio of animals that leave the bacteria alone. So next, we ask whether this bacterial avoidance forms a really aversive memory. To test this, we first test whether animals develop a specific aversive memory to the bacteria. We trained animals with concurrent mitochondrial stress on either on OP50 or HT115. These are two different innocuous bacterial strains with different cues, which animals originally like. After training them for 3 hours, we transfer animals to antimycin-free plate with different innocuous bacteria that they originally like. Of note is that because higher dosage of antimycin that we used earlier will impair the locomotion and viability after wash and transfer. So therefore, in the following experiments where transfer animal is necessary, we use lower concentration of antimycin to preserve animals' viability and locomotion. And we found that animals display higher avoidance toward the bacteria they are trained with. So as you can see here, if animals are trained with OP50, then it shows higher avoidance when they transfer to the antimycin-free plate that contain with OP50 compared to HT115 and HB101, which is two other bacteria strains. On the other hand, if they are trained with HT115, then they show higher avoidance toward HT115. So these data suggested that there's like a specificity for this aversive memory. So next, to substantiate our hypothesis, we developed another choice assay to confirm the aversive memory. We train animals on OP50 with antimycin and allow them to recover at another bacterial strain HT115 without anti-mycin for various durations and test their preference toward OP50 for OP50 in a bacterial chemotaxis assay. So we calculated chemotaxis by the ratio of animals that choose OP50 over the LB. Therefore, the positive value indicated that animals like OP50, while the negative value indicates that animals avoid the OP50. We found that animals formed a aversive memory toward OP50 if they are trained with concurrent mitochondrial stress, as shown here. Moreover, we found that the memory lasts at least 15 hours, and animals erase this memory after 24 hours. So therefore, this study, in a further study, we use 15 hours as resting standard protocol for this assay. Also, we show that animal only forms aversive memories specifically to the bacterial strain they are trained with. As shown here, if they are trained with OP50, they only form that aversive memory toward OP50, but not HT115, and vice versa. So here we show that this mitochondrial stress-induced learned bacterial avoidance has that feature of long-term aversive memory, because 15 hours for C. elegans is kind of long. So previously, the Ruvkin lab has demonstrated that sensory input is critical to induce avoidance behavior from various types of cellular homeostasis disruption. So here, to confirm whether sensory inputs are critical for mitochondrial stress-induced avoidance, we first check TEX2 and TEX4 mutant. TEX2 and TEX4 compose a critical cyclin GMP-gated channel that acts downstream of various sensory receptors of different kinds of sensory neurons in C. elegans to activate the neurons after they experience the stimuli. So we found that avoidance behavior is nearly abolished in TEX2 and TEX4 mutant, so suggesting that sensory input indeed are very essential to promote this avoidance behavior. Next, we conduct a small screen of different types of sensations necessary for this behavior by checking mutants by checking mutants strains in which mutations will perturb a specific sensory neuron. For example, we examine GPCR mutants or some receptor type guanylyl cyclase mutants that disturb a specific type of sensation. Or we also examine the terminal selector mutants that some specific sensory neurons are not fully developed. So to make a long story short, we found that olfactory neurons AWA, AWB, and AWC, and gustatory neuron ASEs, and the gas sensing neuron BAG are required for this aversion. So ASE neurons are pretty much interesting because they are functionally left-right asymmetric. The left ASE neurons sense the increase of salt, while the right side of the ASE neurons sense the decrease of the salt. So we wonder whether this left and right ASE plays different roles in this behavior. To test this, we use left and right specific promoter to kill ASE to answer this question. We first confirmed that killing both ASEs will reduce the avoidance behavior. And interestingly, we found that when only killing the left side of the ASE, but not the right side of the ASE will affect the avoidance behavior, suggested that the sensory input from the ASEL, which is the left side of ASE, is important for this aversion. And another interesting neuron we discovered is the gas sensing neuron BAG, because BAG sense both decreased oxygen and CO2 presence via different receptors on their membrane or in their cytosol. And in previous, in our screen, we realized that only mutations of GCY33, but not GCY31, will affect the avoidance behavior. And when we dig into literature, we found that in previous study, GCY31 and GCY33 has been shown to play a major role in oxygen sensing, but GCY33 has a minor role in CO₂ sensing. We therefore hypothesize probably CO₂ sensing can be critical. So we checked another mutant, which is GCY9 mutant, in which particular CO₂ sensation in BH neuron is abolished. We found that two independent GCY9 mutants show similar suppression of bacterial avoidance behavior to that of the single mutant of GCY33. Furthermore, double mutant of GCY33 and GCY9 did not further show similar suppression level compared to all the singles, suggested that these two receptors act in the same pathway. So to further substantiate that really CO₂ sensation is required for this behavior, we try to remove the atmospheric CO₂ in the assay plate. So how do we do that? We put a soda line on top of the assay plate, and so a previous study has shown that soda line is efficient to remove the atmospheric CO₂. So in this setting, we found that removing CO₂ significantly decreased the avoidance behavior. And furthermore, it's not further mutation of GCY33 did not further suppress this effect by soda line, suggested that CO₂ sensation is conducted via GCY9 and GCY33 in a BH neuron in this avoidance behavior. Then we next sort of want to ask whether olfaction, gustation that we previously identified is important function together or separately with gas sensing to convey the sensory information. So to test this, we test the mutants that affect olfaction or gustation respectively under soda line setting. Interestingly, we realized that avoidance behavior is not further altered in olfaction defective strain under soda line condition, which suggesting that olfaction and CO₂ sensation act in a similar pathway. While the avoidance behavior is further decreased in the mutants with defective gustation, indicating that gustation and CO₂ sensation act in a distinct pathway to convey the sensory information. So next, we are interested in understanding whether downstream interneurons are involved, because interneuron oftentimes is critical to process sensory information and perform decision making for the behavior outcome. So to reveal interneurons that mediates this behavior, we conduct another targeted small-scale screen again, and find multiple possible interneurons that can be involved in fine regulating the behavior. Here, I'm going to show you two of them. The first one, it's a salt octopamine releasing neuron, RIC. We focus on octopamine because it's closely related to norepinephrine, and octopamine is often considered a major fight and flight neurohormone of invertebrates. And we found that mutations of the genes critical for octopamine synthesis, which is TPH1, significantly reduce the avoidance behavior. So to further validate the result, we use pharmaceutical method to inactivate the RIC specifically. In brief, we use this, we express this histamine-gated chloride channel in RIC. Therefore, if we provide histamine in the assay plate, it will activate this channel, and promote the chloride influx into the channel to hyperpolarize the neuron, which make the neuron more inactivated. And notably, this effect can be reversed quickly after remove the histamine. So here we perform the avoidance behavior in the histamine place to inactivate the RIC neuron. And we found that indeed the avoiding behavior is reduced upon RIC activation, suggesting that RIC activity is critical to promote this avoidance behavior. And the second neuron, interneuron, we are interested in is the AIY interneuron. Why? Because AIY is synaptically connected with various sensory neurons, including the olfactory neuron AWA, AWB, AWC, the gustatory neuron AAC, and the CO₂-sensing neuron BAG that we just identified to be essential. So using strains with AIY ablated or genetic ablation by mutation of a terminal selector TDS3 showed that a rare avoidance in these animals. And notably, we have confirmed that ablation of AIY didn't affect animals' locomotor ability and didn't affect the normal odor preference. So also to further substantiate that AIY activity is required, we use the same histamine method that I just mentioned. And you can see that suppression, inactivation of AIY also suppressed the avoidance behavior. So this suggested that the AIY activity is crucial for this behavior. So now we have two interneurons that enroll function to control this behavior. We next aim to understand whether the activity of interneuron we identify is required at a specific stage for the animals to form this aversive behavior. To test whether neuron is required during memory formation stage or memory retrieval stage, we utilize the fast efficacy and the reversibility of the histamine-gated chloride channel that I just mentioned. We predict that if a neuron is crucial only in memory formation stage, then if inhibition of the neural activity of that neuron at the training phase, but not the testing phase, will affect the behavior. And we found that animals are more attracted to OP50 in this choice assay and showed reduced avoidance behavior in this transfer assay if RIC neurons is inhibited in the training phase. And we didn't see such effect when RIC neurons is inhibited in the testing phase, which showing here, there's no effect compared to the sibling in both assays. So therefore, our data suggested that RIC is required for memory formation. To further ask whether RIC activity is sufficient to enhance the memory formation under the concurrent mitochondrial stress, we use optogenetic method to increase RIC neuronal activity. So here, we use a red light activated channel-rhodopsin, which is called crimson, to do so. Basically, this channel-rhodopsin will be activated in presence of all transretinal and also in presence of the red light. And this activation will cause the influx of cation, which will promote the depolarization, which is activation of the neuron. We found that activation of RIC in the testing phase, but not in the training phase, but not the testing phase, will promote this avoidance behavior. And this effect is pretty specific to the bacterial species, since if we train and activate the RIC on OP50, but perform the testing in HB101, which is another strain, we did not see this like promoting avoidance effect. So here we demonstrated that RIC is both required and sufficient to form aversive memory upon mitochondrial stress. Next, we test the role of AIY activity in the memory formations and retrieval. So basically using the same histamine gated chloride channel method, we found that animals are attracted to OP50 in the choice assay and shows reduced avoidance in this transfer assay only when AIY is inhibited in testing phase, but not the training phase. So this data suggested that AIY functions in retrieving the memory during the aversive learning upon concurrent mitochondrial stress. So far here, I have shown you that animals change their preference toward innocuous bacteria with concurrent mitochondrial stress in the peripheral tissue. And this behavior is governed by olfactory neuron AWA, AWB, AWC, gustatory neuron AAC, and CO₂-sensing neuron BAG, and the two interneuron RIC and AIY. And RIC is critical for memory formation, while AIY is critical for memory retrieval. So next, we want to ask whether RIC function upstream of AIY to convey the memory formation, and where the sensory formation functions. So to test this, we express a gain of function PKC1 specifically in RIC, which potentially constitutively activate RIC neuronal output. We found that in control conditions, constitutively activate RIC does not induce the guideless bar, does not induce ectopic avoidance behavior in absence of mitochondrial stress. However, under mild mitochondrial stress, which we induce by lower concentration of anti-mycin, animals with constitutively activated RIC perform higher avoidance behavior compared to their sibling. And this effect is abolished in TBH1 mutant, suggested that consistent to our previous result, RIC release octopamine to promote this behavior. And interestingly, we found that mutation of Ki1 and TTX3, which cause non-functional ASCL and AIY respectively, also suppress the effect of RIC activation. So suggesting that AIY and ASCL function downstream of RIC. So similarly, we use the same method to activate AIY, and it's pretty much the same like in condition without mitochondrial stress. This AIY ectopically activation does not contribute to ectopic bacterial avoidance. Only when mitochondrial stress, in presence of mitochondrial stress, it will promote the bacterial avoidance. And we found that mutations of Ki1 suppress the effect of AIY activation, but mutations of TBH1 does not suppress this effect. So combining with the result of RIC activation, we favor the model that RIC function upstream of AIY to form the memory via releasing octopamine. And AIY retrieve the memories and induce the avoidance behavior. And on top of that, the sensory information from ASCL is critical for AIY to properly retrieve the memory and generate the avoidance behavior. So next, we wonder how octopamine functions to modulate the circuit. So to answer this, we test which putative receptor is involved in generating this behavior. And basically, we found that mutations of SIR3 and OCTR1 show no or pretty marginal effect, while mutations of SIR6, which is the putative octopamine receptor shows significant reduction of avoidance behavior, suggested that SIR6 here is the octopamine receptor for mitochondrial stress-induced bacterial avoidance behavior. So where does SIR6 function? To investigate this, we first clone the promoter of SIR6 and check the expression pattern. Interestingly, we found that SIR6 expressing ASEL, AIY, BAG, and RIC itself. To test the neuron of action for SIR6, we performed cell-specific rescue experiment. First, we found that restoration of genomic SIR6 by SIR6 promoter itself can restore the avoidance defect in SIR6 mutant. And here we found that restoration of SIR6 in AIY, ASEL, but not in BAG, rescue the defect of SIR6 mutant. So this is consistent with our previous result that AIY and ASEL function downstream of RIC. So how does RIC form the aversive memory? We hypothesize that RIC increase octopamine production to respond to the peripheral mitochondrial stress. So to test this, we investigate the TPH1 gene transcription by promoter assay. Basically, we use TPH1 promoter drive entry fluorophore and measure the fluorescence intensity to indicate the transcriptional activity of this gene. And we found that TPH1 transcription activity in the RIC increased over time upon mitochondrial stress. So indicated that increased TPH1 transcription in RIC after mitochondrial stress. So we next turn to investigate the RIC neuron activity. And we use GChem6 to do experiment. So this part of experiment is done by another PhD student in the lab, Victor. Just give a brief introduction to GChem6. GChem6 is a genetically encoded calcium indicator, which composed of GIP, calcium sensing co-modulating domain, and the M13 domain. When the neuron gets activated, which indicated that a calcium influx into the cell, the calcium will induce conformation change of co-modulating, and this conformation change will result in reconfigurations of the GIP, which make the GIP active. Therefore, we can measure the GIP intensity change to indicate the neuronal activity. And to do this experiment, Victor used the microfluidic device. An animal will be stuck in this chamber with their nose tip exposed to the outside, and then a control buffer or the OP50 supernatant, which is the bacterial Q, will flow by the nose tip. And in the meanwhile, he will record the GIP intensity real time. And we found that bacterial Q induce strong RIC calcium transient in RIC exon when they are treated with antimycin compared to the naive animal. And Victor then carefully dissect the onset of RIC responsiveness. He found that RIC start to respond as early as 30 minutes after antimycin treatment, when avoidance behavior and the TPH1 transcription are not yet developed. And this response increase over time, as you can see here. Since we previously showed that RIC is required for memory formation, we wonder whether RIC still responds to the bacterial Q after training. Therefore, Victor trained the animals with antimycin for 3 hours and rest them on the antimycin free plate. And he found that after resting for 3 hours, some animals still show quite strong RIC response toward the bacterial cue. However, this response is completely disappeared 15 hours after 15 hours of resting. But keep in mind that 15 hours, animals still retain the aversive memory. So this RIC response is nicely coherent with the role of memory formations for the RIC. And Victor realized that dysfunction of downstream neurons ACL or AIY does not affect the response of RIC toward the bacterial cues upon concurrent mitochondrial stress. This further reinforces our model that RIC acts as upstream to respond to the peripheral mitochondrial stress and form the memory at this circuit. But interestingly, Victor found that in the sensory deficit mutant, especially the olfaction deficit, RIC response toward the bacterial cue is nearly abolished. This suggested that olfaction is critical to activate RIC in this mitochondrial stress-induced response. So then we asked, how about AIY? But when Victor looked at AIY response to a bacterial cue, we are sort of surprised that there was no stark difference between trained and naive animal. However, we do realize there is a decreased onset latency in a trained animal. But interestingly, Victor found that the proper RIC responsiveness requires ACL and the octopamine signaling, because RIC responsiveness significantly decreased in KY mutant, whose ACL is not fully developed, and in the SIR6 mutant, where the octopamine signaling is abolished. So this here indicated that RIC responsiveness to the bacterial cue depends on ACL and the octopamine signaling. So last, we try to analyze the behavior strategy of animals in response to bacterial cue in the chemotaxis assay. Animals basically use a strategy called pirouette, and that it's basically a high angle turn to adjust their movement toward or away a cue. We use bearing angle here to indicate the moving direction of the animal. Basically, if the bearing angle of an animal is 0 to 90, it means that animal is moving toward that sensory cue. On the other hand, if it's 90 to 180, which is this way, meaning that animal is moving away from the sensory cue. So if the sensory cue is attractive to the animals, then when animals moving toward the sensory cue, which is the bearing angle is 0 to 90, they will perform lower frequent, there will be less chance for them to perform this pirouette, which is a high angle turn that direct them to the other direction. However, if this cue is attractive, but their moving angle is 90 to 100 and 180 degree, that means they are moving away. So the animals will try to increase this pirouette strategy, which is high angle turn, to redirect their directions toward the sensory cue. So Chien, the brilliant master student, developed an algorithm to calculate the frequency of this high angle turn in the animals. We literally videotape it, recorded it, and analyzed at every point of the this bearing angle and analyze the frequency of the animals that perform the purate. So consistent to my previous, hopefully understandable explanation, if in the naive animal, which they are attracted to OP50, so if their bearing angle is 0 to 90, they show pretty low frequency of purate. And if their moving bearing angle is 90 to 180, they show higher purate frequency. And however, upon concurrent mitochondrial stress, which animal develop aversion toward OP50, the trend is fully reversed. And this reverse trend is abolished in octopamine deficient animal and AIY deficient animal, implying that octopamine and AIY enable the learned avoidance behavior by modulating this purate bias when animal navigated their sensory environment. So here, to summarize what I have told you today, we showed that the state of systemic mitochondrial stress in C. elegans is in part encoded by octopamine circuit. I see likely response to the peripheral mitochondrial stress at early stage and release octopamine to modulate AACL and AIY and form the aversive memory. AIY then integrate various sensory information, including olfaction, gustation, and CO2, and retrieve the memory to direct downstream into motor neurons and direct the purate strategy and to perform the avoidance. So with that, I would like to thank all the members in the Pen Lab and thank Peter for being such a good mentor. And of course, my brilliant student, Jen, to literally complete all the story. And Victor, who complete all the calcium imaging, and Cheryl, who do some revisions for us. And I would like to thank all the first generation and the later generation Pen Lab members for creating such a good environment to play with and study with. With that, I would like to thank you all for listening and happy to take any questions.
Thank you so much, John. That was fantastic. While we are waiting for people, just in case it's a free discussion platform, so if you have questions, feel free to unmute yourself or raise your hand. And okay, we have one question already by Jerry. And Jerry.
Yeah, it's a very beautiful data. Thank you so much. So I'm wondering, because you are talking about bacterial avoidance, so what do immune cells play? What roles do they play in this situation, and how do they interact with neurons in C. elegans? Thank you.
Okay. Basically, we have the data here that normally when the mitochondria stress, when the mitochondria got perturbed, the C. elegans will mount a stress response, which is called mitochondrial UPR. And these mitochondrial UPR act through a pretty critical transcription factor, ATFS1. And by other control studies and the data I show here, we pretty much think that there's two different separate mechanisms, which is upon mitochondria perturbation, they will activate ATFS1, which is more like activated the downstream or the immune response or the heat shock, the chaperone response in C. elegans to combat this perturbation. And the other response is through navigating the avoidance behavior to avoid this mitochondrial perturbation, because here we show that in this ATFS1 mutant, the avoidance is completely not affected, suggested that these two are sort of distinct pathway. So you can imagine that animals will, on the other hand, activate their like responses to combat with this mitochondrial stress, and on the other hand, they will just try to avoid this source to try to diminish the damage.
And how does the RC sense the mitochondrial stress? What's the injury signal?
Oh, then I would suggest you guys stay tuned with our publication. There's one project that literally investigated what's the signal. So I will believe it's soon to be published. So just stay tuned.
Sounds great. I actually have two questions. The first question is really about this memory formation. I found it fascinating that RIC is only involved in the formation, and the AIY, if I remember correctly, is involved in the retrieval. So then I have to really ask this question, then how do you think the memory is, how is and where is the memory stored? Especially this 24 hours period length is very interesting.
I would say it's quite hard to like we did not investigate the the detail, whether like the cellular NMDA receptor or something, like all the cellular things. But yeah, I would say like it'll be like this octopamine release was sort of remodeling the responding neurons and sort of change not only their responsive property, probably cellular changes, because stress-induced is a GPCR, and GPCR can work as a little bit like long-term memory-ish thing. So probably can like affect downstream protein synthesis or something else that need to be looked carefully. So if follow-up studies look at how like AIY shows different protein like response, like how SIRS6 activate AIY and make AIY produce probably different receptors or different synaptic for more synapse or something, then that can probably answer your question.
Do we have any questions? If not, if I can just quickly follow up one, I just found it fascinating that how the functional circuit composition is very different from, or maybe the point I'm trying to get to is you can see the SIRS6 is expressed in many of the neurons that are involved in this avoidance behavior, but somehow that through octopamine, the engagement only require ACE and the AIY, but not the BAG. We're just curious about just in general, like the selectivity or the composition of the functional circuits, like in your opinion, how is formed? Why? I believe these neurons are probably all well connected, or at least through this behavior. I don't have answer for this question myself. I was just as a neuroscientist, I was always curious about how the functional circuit is formed.
I mean, yes, that's an interesting question. Like why the BH neuron express SIRS6, but it's not targeted by RIC. I personally don't have the answer for that, but probably it's the sensory information that provided by BAG. It's another, because if you remember, ASCL and BAG function in that soda line assay, they function separately. So probably AIY like so that means like AIY weight differently from their input. So probably this doesn't require the BAG activation from the octopamine. I have no answer for this, but yes, I was like, because like basically they are like two distinct pathways. So I would believe, yeah.
That's interesting. Thank you so much for giving a wonderful talk today, and we really enjoyed it. And we will stay in touch and to follow up with you. And thank you so much for everyone joining us today. And let's just give another round of applause to John. And hope you have a great weekend.
See you.
Bye bye.